Great. Welcome, everybody. You're at Jimby Live. This is a free webinar series offered by the ASM Education Department. My name is Rachel Horak, and I'll be your host for today. Today's speakers are Dr. Tess Killpack of Salem State University and Dr. Sarah Fulmer of the University of Guelph, and I'm so happy to have them here today. Into the chat box, I'm going to put the link to their paper that you can read open access if you haven't already. And the title of their paper is Increase Scaffolding and Inquiry in an Introductory Biology Lab, Enhance Experimental Design Skills, and Sense of Scientific Ability. The format of today is they're going to give an overview of their presentation of their paper for about 20 to 30 minutes. And feel free to use the Q&A box to add questions. And after their short talk, they will come on and we will have an interactive Q&A using the questions that you submit through the question box. In addition, you're welcome to use the chat box to converse amongst yourselves. If you have technical difficulties today, you can send a direct message to Justin Van Blaricom, listed as 2.Justin Van Blaricom, and he can help you with audio or visual issues today. Okay, so with that, I'm going to turn it over to our speakers. Thank you so much for being here.
Okay, I'll get my screen going. Sarah, can you see that all right? Okay.
Yeah, we're good.
Thanks everyone for being here and happy Friday. I want to thank Rachel and Jimby Live for inviting us, and also want to acknowledge our publication co-authors, Julie Roden, Jocelyn Doltz, and Krista Scow, who contributed to the course redesign that we'll talk about and to the publication. So let me get my mouse going. So my name is Tess Killpack. I'm an assistant professor in the biology department at Salem State University, which is a regional public institution just north of Boston, Massachusetts. And my primary role there is to teach. I teach many flavors of introductory biology and also do some research on biology teaching and faculty professional development.
Hi, everyone. My name is Sarah Fulmer. I am an educational developer in the Office of Teaching and Learning at the University of Guelph in Ontario, Canada. My primary role there is to support faculty, instructors, and graduate students with their teaching, and also to work on the scholarship of teaching and learning and disciplinary-based educational research with faculty and instructors. This work was done while both Tess and I were at Wellesley College in Massachusetts, and Tess will talk a bit more about Wellesley College in a little bit, but since then we've both branched off to different institutions. So we'd like to get to know you and who's in our audience today. So if you feel comfortable, we'd love to know what your role is at your institution, and some topics, some keywords that you'd like to discuss or learn about in today's session. So please take a moment, and if you feel comfortable, type in the chat.
Hi, Julie.
Hi, Julie.
And everybody. Hi, everybody.
Okay, so please keep typing in the chat as we move on and see who else might be here.
Okay. So what our paper presented and what we're going to talk about today is a model for redesigning a lab curriculum, specifically focused on introductory level undergraduate biology labs. The course that we redesigned was one half of an introductory sequence, the organismal sort of ecology, evolution, organisms half of the intro bio at Wellesley College. Wellesley College is a small, private, highly selective women's liberal arts college that's just outside of Boston, Massachusetts. So the paper goes into a little bit more context sort of about the institution itself, and I'm happy to share more about that if you have questions. So the premise for this research study really came from sort of a pedagogical and curriculum desire to redesign the laboratory course to increase opportunities for students to engage in meaningful inquiry, and also to increase student recognition that they are developing really concrete and transferable skills of scientists. So we, as instructors in the department at Wellesley who taught the course, recognized that these were some areas for improvement. And we also knew from the literature that enhancing inquiry, enhancing student ownership has a lot of benefits documented in terms of their sense of self-efficacy and science identity and persistence in STEM fields. So with these redesigned goals, we also decided this is a great opportunity to do a research study. So the research goals that we had associated with this were to examine the influence of our course redesign on student gains in experimental design skills, students' awareness of their own skill building, and students' perceptions about the relevance of their laboratory course research experience.
So we'd like to get to know a little bit about what you teach if you are an instructor, and it looks like from the chat that a lot of you are instructors. And so these are 4 areas of skills that were highlighted in the lab curriculum for this particular course. So we'd like to know if you teach any of these 4 skills and which ones you teach. And the second question that we'd like to ask you is, which of these skills do you think is the most challenging for your students to learn or to master? So I've copied a link to a Google form into the chat. So if you feel comfortable answering these questions, please click on the link to the Google form and respond to those questions, and we will look at the results together. Oh, sorry about that, folks. Thank you for letting me know. I will put this link into the chat one more time. Hopefully that worked for everyone. And if you don't teach, but are hoping to teach at some point in your career, then answer these questions as if you were teaching your ideal course. So we see some results coming in, which is good news for us that the link is working. So please take a moment and fill out the survey if you're still looking at it. Yeah, if you're a student, we would love for you to fill out the survey in terms of what skills you're being taught and which one you find the most challenging to learn. This is great to have multiple perspectives. Okay, so in terms of results that are rolling in right now, we have about, I think, 56 responses so far. It looks like all 4 of these skills are addressed in most of your teaching in some way. These are key skills for biology students and science students to learn. So I'm not surprised that most of the instructors here are addressing those skills across the board. I think the one that seems to have the fewest responses is designing inquiry-driven scientific experiments. So this was a key feature of our course redesign, which made it quite unique and distinct from previous iterations of the course. Looking at our responses to the second question in terms of which of these skills you think is most challenging for students to learn, the most common response here was designing their own experiments. So again, this is a very challenging skill for students and for us to learn as researchers. And so I'm not surprised that that's the most challenging. The one that had the least number of responses was collaboratively making observations and measurements. So what we're going to sort of launch into now is Tess will talk about the redesign and how these 4 skill areas were addressed by course changes that were made.
Okay, my screen share is back on, just to make sure I'm not. Okay, so I want to provide a bit of an overview of sort of how we redesigned the course in terms of weekly activities, and then we'll get more into the scaffolding. So as I said, our team of instructors decided that we wanted to redesign the laboratory course pretty much the entire semester. And what we did is we had funding over a summer to really take time to articulate learning objectives for the course in a very careful and systematic way, and also to design iterative and scaffolded skill building across the semester. So some of the key ways that our redesigned course differed from the original format were that in the original format, there was the first week was just an observational lab. Students collected water, learned how to use a microscope, and looked at plankton under the microscope. So what we did with the redesign is we created instead an introductory module that lasted 2 weeks. And in this, we were still having students work with plankton and use microscopes, but we were actually having them collect quantitative data and test some hypotheses. So introducing inquiry right from the start and having students generate results that were unknown to themselves and to instructors and start learning some analytical techniques. Another really big difference between the original course format and the redesigned format was creation of what we called the capstone module, which came at the end of the course. So in the original course format, students did design a group experiment, but the way that the experiment was set up was it was a population growth experiment using flower beetles. So starting right off the bat in week 2, students were setting up their group experiment without having any instruction or practice in experimental design at all, because they had just that first observational lab before they were setting up their experiments. And then the flower beetles were put in an incubator, not looked at again until the final weeks of the course when students analyzed the data and created presentations. So we wanted to more thoughtfully build up to this student designed research experience. So what we did is after the introductory module, we placed a middle module, which had a lot of similar features to the original format related to plant form and function, but had some enhanced inquiry. And I'll tell you more about that on the next slide. There were some dissection labs that were unchanged, but then students used what they learned from the introductory and the middle modules in terms of experimental design and analysis skills to then complete the capstone module, which used a new model organism and involved student designed experiments as well as scholarly presentations. With the change in these modules, we really thought carefully about scaffolding skill building across the modules. So we started with those 4 key skills that you responded about in the Google form, and we broke them out into sort of smaller sub-skills. And then we thought across the 3 modules how we would build and practice those skills. So I'll walk you through this. It's a lot to look at once, and this table is in the paper if you want to take a more careful, close look after the presentation. But in that introductory plankton module, students were collecting and measuring organisms starting in week 1. And they were learning about experimental design in the context of instructor-defined experimental questions and hypotheses. In this first module, they learned how to make graphs of mean and standard deviation and perform 2 sample t-tests, and also got started with some scientific communication skills, learning how to write figure captions to accompany their graphs, and also results text that incorporated their t-test statistics. So these little eyes show where the skill was introduced, and then the shaded squares beyond show that it was practiced in an iterative way in the following week. With the middle module on plant form and function, you can see that students continued to work on instructor-defined experiments. They continued to use the graphs and the t-test that they had learned in the previous module, but were also introduced to an additional statistical test called the ANOVA with some Tukey's post hoc testing. In this middle module, they also were introduced to writing discussion sections of scholarly papers or lab reports, and using literature and citations to support the discussion. And then in this final capstone module, students, a big change was that in this sort of focus on experimental design, students shifted from instructor designed experiments to student designed experiments. So they generated their own experimental questions and hypotheses using our model organism. They defined their experimental groups and designed their experiments. So they could use what they learned in those previous 2 modules. And they continued to use their analytical skills. And with this final module, they were able to practice selecting appropriate statistics and figures based on the data that they collected for their capstone experiment. And finally, we added additional writing skills with students writing descriptive method sections and also preparing and presenting scientific posters.
So I'm going to talk a little bit about the research and study design and the methods that we selected to assess the effectiveness of this course redesign on students' learning. So as Tess has mentioned, there was an original format of the course. This happened in spring 2016, long before any pandemic was in our sights. There were 5 sections of the course, so 5 lab sections were part of the study that were led by 3 different instructors. The redesign happened over the summer, and in fall of 2016, the redesign format was launched to a new group of students. There were 5 sections of lab as part of this study, and the same 3 instructors were leading those 5 sections of labs. In terms of measures that we used, we had measures of students' inquiry skills and students' experiences, and we also had measures of students' perceptions of their skills, knowledge, and their learning. So in terms of the measures that we use to assess students' learning and students' skills, the first one is the tool for interrelated experimental design, which Tess and I published in JMBE in 2018. I've just put the link to that paper into the chat if you're interested in reading more about this tool. The basics of this tool is an assessment of students' ability to design their own experiment. And it comes with a scoring tool, which assesses in a pretty nuanced way what students' skills are in their ability to design their own experiment. So this assessment was done at the beginning of the semester, so a pre-assessment, and at the end of the semester as a post-assessment in both the original and the redesign format. Okay, so we were able to use this tool to detect change in students' abilities to design their own experiment. We also use the LCAS, the Laboratory Course Assessment Survey, as a measure of students' skills. The LCAS was developed to distinguish cures, so undergraduate research experiences in courses, versus traditional laboratory courses. And so Tess will talk a bit more about this later, but this redesign was not meant to be a cure. It was meant to have aspects of a cure without doing the full cure experience for students. So we used this assessment to determine whether students perceived aspects of this lab as having some features of a cure. And that was just done at the end of the semester as a post survey. We had two different measures of students' perceptions of the lab experience. The first one was a personal skills rating on a mastery rubric, which was done at a pre and post at the beginning and end of the semester. We'll show you a bit more about this tool in a later slide, but basically it's just a tool that asks students about their scientific skills in a number of areas in terms of how confident they were in their skills. Unique to this particular research study was we also analyzed students' end of semester course evaluations. So your traditional course evaluations that are done at the end of the semester. At Wellesley College, at this particular institution, those course evaluations are completely open-ended. So there's a series of 3 prompts that ask students about what they thought about the course, what they found valuable, what they'd like to improve. And we analyzed students' open-ended comments as part of this research study. So we'll show you a bit more about that later as well.
So I'm going to walk through some of the quantitative data that we collected, comparing the original and the redesigned format. So the first set of data are from the experimental design assessment. And we wanted to compare growth in experimental design skills across the semester in the 2 different course formats. And the very helpful and generous peer reviewers at JIMBY suggested that we present normalized change as a measure of student gains on the assessment. So we can talk more about this after the presentation, but briefly, normalized change is basically a ratio of the realized improvement to the maximum possible improvement based on a student's individual pre-test. So this normalization allows instructors to compare gains basically independently of students' pre-test scores and can basically do so across many different student populations. And for example, in our case, different course sections and different semesters. So what we found was that students in the redesign course showed significantly higher gains on this experimental design tool. You can see scores in the original format here in gray, and scores in the redesign format were significantly higher for those students. So this shows that this redesign course and this inquiry and this careful scaffolding was correlated with increased gains in experimental design skills. Next, I'll show, these are the results from the laboratory course assessment survey. So in this survey, it asks students to report how often they feel they engaged in collaboration throughout the semester. There are also several prompts about students' perceptions of elements of discovery or relevance of their course research experience, and also elements of iteration, sort of a chance to practice skills repeatedly or repeat experiments if needed throughout the course. So interestingly, students in both the original format, which is shown here in the gray bars, and the redesigned format provided very high ratings on this assessment survey. So for the most part, students felt like they were engaged in collaboration and discovery, that their course experience was relevant, that they had chances to iterate in terms of their research skills, whether they were in the original format or the redesigned format. And you know, this wasn't necessarily anticipated. We personally, as instructors, thought that the original format was lacking in a number of these elements, but this perhaps points to that these are introductory students, that perhaps any sort of laboratory experience where they're engaging in some of these skills feels relevant and feels like science to them. This last quantitative tool is the personal skills rating. So this was an internal assessment that we created to really hone in on those key skills that the redesign was focused on. So the experimental design, collaborative data collection, data analysis, and scientific communication. And both before the course and after the course, we had students rate themselves on a mastery rubric, which is shown down here below. So for each of these 4 categories of skills, students rated whether they felt that they were beginning, developing, proficient, or masterful based on the prompt descriptions. And when we compared the data from the 2 semesters, we did see that in both course formats, the original and the redesign, students' personal skills ratings increased. So from pre to post, students' skills did increase. There were significant changes in both course formats. Yet what we did see is that in the redesign course, the post-semester ratings were significantly higher than the original course. So I have the averages from the post-test scores shown here under each of the 4 skills. So you can see for experimental design, students in the original course on average rated themselves as proficient, whereas in the redesign course, we're moving into proficient, masterful. Similarly, for collaborative data collection, we see sort of a significantly higher average score in the redesign course and more movement towards students feeling masterful in those skills. Same for data analysis and presentation. With scientific communication, there wasn't a significant difference in those post-test scores between the original and the redesign format. So generally, students reported that they did feel gains in those skills, and that those gains for 3 of the 4 skills were significantly higher with the redesigned course experience.
So I'm going to talk a little bit about the data from the end of semester course evaluations. One of the reasons we included this qualitative data as part of this research study is we saw some of the limitations of the quantitative data that was being collected, and students' sort of inaccurate perceptions at times of their own skills and their own learnings, especially at the introductory level. So our decision to include these course evaluations was to see, what comments would students have about this lab experience when asked just plain open-ended questions. So what topics would spontaneously come up at the end of the semester when they reflected back on their experiences in the course? So one key area that stood out to us in the course evaluations was related to the authenticity and the relevance of these labs. Students in both the original formats and the redesign format said that the labs were practical and valuable. The key difference here was that students in the redesign format were more likely to mention that they designed or conducted their own experiment. So that's 34% of students in the redesign format commented on that versus 14% of students in the original format. So that skill or that experience stood out to students as something worthy of commenting on. Also in the redesign format, students were more likely to use the words real science or actual science. So it was 15% of students in the redesign use those terms in their course evaluations versus 3% of students in the original format. So again, these sort of the relevance or the experience of these labs really stood out to students as something worth commenting on. Another particular area that really stood out in the course evaluations was students commenting about the skills that they had learned. The interesting finding here was that in the original format, students would comment that they learned scientific skills or laboratory skills or writing skills. So they would be very generic about their comments of the skills they had learned. This was in contrast to the redesigned course evaluation comments, which mentioned very specific skills that students had learned. And so, for example, students in the redesigned format were more likely to mention specific statistical tests that they had used, whether that was a t-test or ANOVA or how to interpret data. 72% of students in the redesigned format mentioned a specific statistical test or skill versus 42% in the original format. This was also true about writing. So students in the Redesign format would comment on things like writing a results text or writing a figure caption, compared to students in the original format who just talked about general scientific writing. So what stood out to us here was that students retained those skills and remembered what skills they had learned and found it worthwhile to mention those specific skills when talking about their evaluations of the course in general. The final area that stood out on these course evaluations were students' comments about their own learning, their own motivation and attitudes towards the course. So we searched for the word learning or learned, and we found that students in both formats talked about their learning on their course evaluations. It was a bit higher in the Redesign, but it was common across both formats. The interesting pattern that we noticed was when students were talking about their interest or their enjoyment or their excitement about the labs. What we saw was that students in the Redesign format were less likely to talk about their interest or their enjoyment in both positive and in negative ways. So all this to say that students in the original format of the course talked more about their interest and enjoyment and excitement. And students in the redesigned course talked less about these feelings or these attitudes towards their lab experience. And we can interpret this in a couple of different ways, and we don't have all the information to interpret this. So it might be that students in the redesigned course liked it less. That might be true. But it also might be that they preferred to talk about other things in their course evaluations instead of just their interest and enjoyment in the course. They were talking about specific skills they had learned, specific experience they had in the lab, instead of perhaps defaulting to just talking about their interest or enjoyment. So lots of ways to interpret this, and it's a really interesting finding that we wouldn't see if we had just used quantitative measures in this case. So I'm going to briefly talk about some conclusions and then pass it over to Tess to talk about some of the implications of this for our teaching. So in general, what we saw was that students in the Redesign format had significantly higher experimental design skills by the end of the semester and more consistent gains in these skills over time. They also thought of themselves as more skilled in areas of scientific writing and statistical analysis and data analysis, and were more likely to mention learning these skills in the course evaluations. And finally, what we saw was that students in both formats thought that these labs were relevant and practical and important, but students in the Redesign format were more likely to feel like they were doing real science.
So some takeaways from sort of pulling this all together and thinking about the implications. One that we came away with was that we saw that with this scaffolded inquiry model, we had really significant gains in experimental design skills over the course of the semester. So this may be indicative that sort of full conversion to what is, I think, often traditionally interpreted in the literature as a true course-based undergraduate research experience or cure, where you have one research project that's usually part of a faculty member or graduate student's body of work, and you sort of work on that for the entirety of the semester. That may not be necessary in order to get some of these really key skills built in students, and that even with this scaffolded inquiry model, students really felt like they were engaged in authentic research experiences. So this has implications because at many institutions, including many that I've worked at, all that I've worked at, there are limitations in terms of supplies and budgets, and there are places where there aren't robust graduate programs or robust faculty biology research programs to really feasibly implement cures. So this is perhaps another way to infuse these elements into courses and get those really positive benefits for introductory students. And then a second piece, and Sarah touched on this, was we were really struck by the value of using these open-ended evaluation questions to gather student voices. So to complement the quantitative data, to complement the more traditional instruments that are out there in our biology education literature, we really found value in gathering these students' voices to assess the impact of the course experience. So not only did we get information about their attitudes, but we found it to be really meaningful that if they were given a blank prompt box, that they could populate it with these really specific skills and practices that they learn throughout the course. And we feel like this indicates a real sense of scientific ownership and a sense of belief in their own skill building and self-efficacy, which we know are important for student persistence in STEM majors. So we can talk about sort of pros and cons of quantitative and qualitative analyses. Qualitative certainly takes a lot more time and energy, but I think it really added value to our study and more insight into what the student takeaways were.
So Tess, I think we might move right on to question and answers.
Sounds great.
Yeah.
Okay. So thank you. And I think Rachel, do you want me to stop sharing my screen?
Yes, stop sharing at this time.
All right. I think I did. Let me know if it's still up there. Thanks, everybody.
Thank you very much. This was an exciting paper.
Thanks.
First, before I get to the questions, I'd like to ask you if you could take just a moment to tell the audience very briefly how you develop the skills and professional development to do such a study and get it published in GIMBY. Did you take special courses? Did you work with some folks? How did you get these skills?
So I can start. I did my PhD in zoology. So I studied bird development. And while I was at the University of Wisconsin in Madison, and I knew in graduate school that sort of my goal with my PhD was that I was really interested in teaching. I felt more motivated to teach students how to do science than sort of commit my life to heavily researching for my own research program and for the sake of making discoveries. So what I did to sort of gain some of this training to kind of start thinking about a scholarly approach to teaching and assessing teaching and thinking about teaching in a scientific way was through a CERTL program, so Center for the Integration of Research, Teaching, and Learning. It's a network of programs that are a number of institutions. So if there are graduate students on with us today, that's a great network to look into while you're doing your scientific training. I had the opportunity to take courses and do a mini research project and learn about how to design research studies. And that definitely stuck with me. It's helped improve my teaching, and I felt like it really gave me a good basis to start sort of diving into writing up journal articles and producing studies, and sort of ongoing professional development through biology education societies like ASMQ and SABR, the Society for the Advancement of Biology Education Research, have definitely helped me continue to hone skills and learn new techniques. And I think in general, sort of a final thing is reading papers, reading about papers in GIMBY and in other biology education journals. You get ideas for the many possible ways to set up research studies and to analyze data. So I'm always getting new ideas from reading papers.
Awesome. Thanks, Tess. I think for me, so I've always been in education. So I was a faculty member of educational psychology at one point in my past career. And since then, I've been working in offices of teaching and learning at various institutions in a faculty development or educational developer role. So my interest has always been in education and students learning and students motivation. And that was my key research area as a master's and PhD student. So it's not surprising that I'm ending up on papers like this, but I think my key piece of advice for all of you is to collaborate with folks in your office of teaching and learning. So In this particular collaboration, Tess brought me on to her research team because I had skills in qualitative data analysis, in the assessment of student learning, in understanding students' motivation, which are skills that she didn't yet have on her research team, but needed to support this project. So through the collaboration between us, we were able to build this research design and this research study. She brought in the course design expertise. I'm not a biologist. I don't know anything about biology education, but she had that expertise with her other co-instructors. And I brought in the sort of scholarship of teaching and learning, research design, and analysis. So that's my recommendation for all of you, is to find folks who have the strengths that you don't, so that you can conduct a research study like this.
Great to hear you say that. This is the 7th or 8th webinar in Jimby Live, and I've asked everybody the same question. And find collaborators is always part of somebody's answer. That's a great lesson.
Great.
Okay. So one of the most popular questions answered by one of your co-authors, Julie Roden, in the Q&A, was this course integrated or tied to a lecture course? And just in case people didn't see it, this was a lecture course attached to labs that students co-enroll in. So that was a very highly rated question. Next question, how much time did the redesign take? And how was this effort supported by the department?
That's a really great question. So we did the bulk of the redesign work over the course of a summer. And it was a team of 4 laboratory instructors who all, and Julie Roden is on, she was one of them, to really conceptualize sort of the course overall, and then kind of divide and conquer to pull the elements together in an interconnected way. So in addition to our team of lab instructors, one really cool thing that we did was brought on 3 student researchers to join us in the redesign effort. So in addition to our instructional team, we had a team of 3 students who helped us pilot these experiments to make sure that they felt relevant and interesting, and they generated a lot of preliminary data. So that took the whole summer, and then admittedly sort of into the fall, we were, pulling things together because it was a really substantial overhaul of the course. We were supported by funds from a Mellon institutional grant that supported redesign efforts and sort of course-based sort of assessment efforts in general. And we also had some departmental support. So we were lucky to have some stipend support to pay us to do this work over the summer. And I would highly recommend that you, as much as possible, can seek those opportunities, because so often sort of instructors take on, uncompensated labor that's really substantial and really improves the departmental curriculum and student outcomes. So we were lucky to have those funding sources, as well as funding for the students to help us with that work.
Was there consideration for the difference between the autumn and the spring students?
That's a great question. Yes, and in the paper, we do provide some data. I believe it's in the supplemental information to just sort of show a breakdown of the students in the original and the redesign course sections in terms of what year in school they were, what their major was, if they were declared major or undeclared major, and if it was biology major or another STEM major. And we also took into account whether students were taking their very first introductory course, or they were taking our course as their second introductory course, because at Wellesley you can take the cells and the organism courses in either order. So we did take those considerations into account. The data are in the paper if you want a closer look at all of the actual numbers, but we didn't have any significant differences in terms of biology majors in either semester, number of students who were on their second intro course versus their first intro course, or students at different sort of class levels in terms of first year, sophomore, junior, senior. So that's a great question.
I think one of the other things that we tried to do and could do better, given, designing this again, was using assessments that had a pre-measure as well as a post-measure. So we could see where students were starting out, and if, particular lab sections or semesters had students that started out really high on skills or perceptions of their skills, then we would know that going in versus just using measures at the end of the semester.
Thank you. So, did you ask the students what inspired their experimental design that they did themselves? Was it maybe web searches or curiosity or previous experience with other models, such as those used in the class?
Great question. So with the capstone module, we had some constraints in terms of the experiments that students could design. So all students had to use the model organism, these adorable little cabbage looper caterpillars. So everyone had to design an experiment using the caterpillars. And with the help of those research students that we had during the summer, we had them help us generate sort of different categories of experiments that are possible with the model organisms. So looking at general sort of competition among organisms, looking at food types, but students could sort of, as long as they were using the cabbage looper caterpillars and designing response variables that could be measured in the lab, they were left pretty open to what they were allowed to design. So they were constrained in terms of the experimental organisms we had and the equipment we had to measure responses from the organisms in the time that was available. But beyond that, the students could work in their small groups to design an experiment that was interesting and exciting to them. In addition to providing sort of the organisms and the possible equipment or response variables to measure, we, as part of the redesign work in the summer, we pulled together a set of journal articles that were related to herbivory, so plant eating in insects and competition in insects, so that students could design their experiments with a literature basis. So they could comb through those experiments, get some inspiration, ask some follow-up questions from those journal articles as a jumping point for their experimental design. And they were also encouraged to find articles of their own that were available.